All right, so we're going to talk about blood now. Blood is the only tissue in the human body that's a fluid. And so it's also classified as connective tissue. And it's actually made up of both living cells and non living matrix. And so the non living matrix is actually the blood plasma, which includes things like water, salts, proteins, antibodies, all sorts of things that can move around in the plasma. In fact, your kidneys actually filter the blood plasma, which then becomes urine. So your waste products like urea is found in the blood plasma, as well as albumin, and other types of proteins. Platelets, all sorts of things will be found in the floating around in the plasma. Again, the living cells will be your red blood cells, also known as erythrocytes, as well as white blood cells like leukocytes and so forth. The blood is typically a dull red color to bright red color. Some people think it's blue, but that's just because of what it might look like through their veins. But realize you're looking through skin and there's lights factors and all sorts of things that make it appear blue, but your blood inside your veins, even though it might look bluish on your skin, is actually red. And that is due to hemoglobin that's present in red blood cells. Of course, it has an iron group with it, and the oxygen attaches to it. Or in a worst case scenario, as I mentioned earlier, carbon monoxide might attach to it, and it actually would hold on to it more strongly, as I mentioned in the previous lecture, than actually oxygen does. The pH of blood is around 7.35 to 7.45, and it has to remain in this stage. If it goes way beyond, or a little bit beyond this, it's actually enough to harm you and kill you. And so there's lots of buffers also floating around in your blood to keep your pH in this critical range of survival, and thus homeostasis. The blood temperature is actually slightly higher than the body temperature. So your body's actually a little cooler than the actual blood that's floating around it. And blood is important in helping regulate body temperature as well. In extreme cold, the body will try to shunt more of that blood towards the inner core. And when you're hot or warm, the blood and the capillaries will dilate to help cool you down. As I mentioned before, the fluid portion of blood is made up of plasma. And again, it contains gases like carbon dioxide, but usually in the form of bicarbonate. Again, that's going to be important for helping buffer blood, along with a variety of ions. Remember, ions are just atoms with electrical charge, like cations, like sodium or chloride, which would be an anion. So actually, blood has kind of a salty taste to it. There's also a variety of proteins like albumin that's floating around in it, as well as hormones. So blood is also very important for transporting hormones, let's say from your testes to other portions of your body that would have receptors that respond to it, or estrogen, or just a ton of different types of hormones out there. Blood is also important for transporting nutrients such as glucose, amino acids, lipids, lactic acid, and cholesterol. As I mentioned before, circulating proteins include albumin, along with antibodies, hormones, and various carrier molecules. So these are proteins that help maybe to move lipids around, like lipoproteins. And if you've heard of high density lipoproteins, HDLs and LDLs and stuff like that, low density lipoproteins, these are examples of proteins and carriers and so forth. The plasma is a similar composition to tissue fluid. That's why it's considered a liquid tissue, but it does have a higher concentration of proteins. So if you were to draw your blood and place it into a centrifuge that would spin, you would get what we call a hemocrit. And then we actually call them hemocrit centrifuges. Often these are glass capillaries that you can fill up with blood. And you spin it for a little bit, and you'll see that the heavier portion, the blood cells, will be at the bottom, and the blood plasma, the watery portion, will be at the surface because it's lighter in weight. And then I believe in the middle, between the two, you'll see Maybe in addition to some lipids and stuff like that, you may see some other types of blood cells and fragments like platelets. Now, the blood plasma represents about 55% of your so the watery areas represent about 55%. In fact, if it ends up being more like 60 or 70%, then that's a good example of someone who is anemic and not making enough blood cells. And actually, if you're dehydrated, you might end up with 50% or something like that. But again, water is the solvent that's carrying the other substances. And then you're going to find all sorts of ions or electrolytes like sodium, potassium, calcium, magnesium, biocarbonate, which is the way to transport carbon dioxide, that also is important for buffering. So there'll be other buffering agents. It's important for maintaining osmosis balance. Osmotic balance is important, and the water is important for that. So it'll help draw water out or salts out of your tissues and so forth. As you understand, osmosis is the movement of water across a semi-permeable membrane. Plasma proteins include albumin, fibrinogens, and globulins. The fibrinogens and globulins, particularly fibrinogens, are important um, for when you have clotting factors and your blood's clotting. Um, Again, there's going to be a lot of antibodies running around and defense, or for, as you understand, antibodies are going to attach to antigens like maybe found on the surface of a virus and help your body to get rid of things that aren't supposed to be there. There's a variety of substances in your blood, like glucose and vitamins and so forth, as well as waste products. After protein metabolism, you're going to have urea and uric acid. These will be filtered by the kidneys ultimately. Respiratory gases like oxygen, which will be held on at some extent in the blood, but mostly on the hemoglobin, as well as carbon dioxide, but usually in the form of bicarbonate. And again, a variety of hormones. The cell types, or the cells that you'll find present, the vast majority are going to be erythrocytes, which is our red blood cells. You'll see they have kind of a concave shape to them. And they're actually, for the most part, just a big bag of hemoglobin. And again, its job is to transport oxygen and to a lesser extent carbon dioxide. Obviously, getting rid of carbon dioxide is important, but it'll be held in the form of bicarbonate and not attached directly to the hemoglobin. We also have leukocytes that include white blood cells. So this is another word for white blood cells, but again, they're leukocytes is the proper way to refer to them. In comparison to millimeter cube of blood, where we had around 4 to 6 million red blood cells, in regards to immune cells, we have around 4,000 to 11,000 for that same cubic centimeter of blood. And we'll talk about these different ones, but we have things like basophils and cytophylls, neutrophils, monocytes, and lymphocytes. Each one of these different types of white blood cells have a different function. And then we have a lot of platelets running around. Platelets are fragments of blood cells. We have around 250,000 to 500,000 platelets per cubic millimeter of blood. Again, very important, along with fibrinogen, to make blood clots. Here's just another example of the hematocrit. Again, you can see that the middle portion has a lot of the white blood cells. The blood plasma represents over 50%, 55, 58%, 40% something, 45% for the red blood cells, and less than 1% for the white blood cells. And then again, the hemoglobin is going to be found primarily in the red blood cells. And when that hemoglobin is finally broken down, actually, it's what gives your urine your yellow color, or used as bile salts. But again, this is the hematocrit, and that is the percentage of the total blood volume that's packed in the centrifuge. And so these are, again, the relative amounts of each. This is also very important for determining your basic health. Here's more of that. Again, some of this I've maybe already mentioned. Again, the hematocrit is a measure of cellular proportion as a percentage of the total blood volume. we've already talked about the platelets and so forth. So this is just a little bit more of what you'd seen in the previous slide. Again, the plasma is the fluid matrix. Most of the cells in the blood are erythrocytes. Again, they're bioconcave, they're flexible discs, and they're just packed with hemoglobin. In fact, they even eject the nucleus in the formation of hemoglobin. So initially, the cells, the new hemoglobin that's brand new, that's been made in the bone marrow, will actually have a nucleus, but eventually that nucleus is either disintegrated or rejected, and it ends up just being one big bag of hemoglobin. I think lasts about 7 months and then gets broken down, I believe, by the spleen, but I can double check that in a moment. Again, that'll give you your urine its yellow color, and it's also going to be used in helping make bile salts. And if you might, as you'll find out later, bile salts are very important for digestion of lipids. So that's the big thing about bile salts. Red blood cells, again, are going to be generated in the bone marrow, particularly the long bones of adults. And these are generated by stem cells. So a stem cell, as you might remember, is a cell that has the potential to become any other type of cell. Initially, stem cells might be called totipotent, but once they're kind of down a course and they become a particular type of cell, then they're called pluripotent. And so the pluripotent stem cells are going to be kind of designated to become blood cells, then are going to be one of either erythrocytes or white blood cells or something of that nature. Now, what controls the production of blood cells is erythropoietin. It's a hormone that's made from the kidneys that helps your body to make more red blood cells. This would be considered a doping drug if it was used for athletic purposes in a federation that was against it. It can be this drug, erythropoietin, can sometimes be given to people that don't have enough blood cells. Maybe they have a disorder where their body isn't making enough. This might be one way to do so. Another form of blood doping, of course, is just to take it directly from blood donations. Some people even go to extremes in sports where they'll take their own blood, freeze it, or put it into a container that stores it, and then returns it to the body a few months later. Other ways that they can increase their red blood cells is maybe training in high altitudes, or things that simulate high altitudes. So erythrocytes are the salmon-colored bioconcave discs. They're anucleated. That means they don't have a nucleus, and they're literally a sac of hemoglobin. Most of the organelles and the nucleus has been ejected out of the body after the cell has gone through its development. So the nucleus does its thing, and then once there's a huge number of hemoglobins in it, the nucleus is ejected. And so the cell life is about 7 months, and then so there's always a constant need to make fresh erythrocytes. And again, your kidneys are going to be involved with releasing of that hormone that helps to stimulate it. Then there's a lot of different types of leukocytes. There's things like neutrophils. Neutrophils have this, so these are going to be stained cells. They might not necessarily look purple or red with big dots until they've been stained. And so we have some granulocytes here. And so they stain pale pink and contain fine granules. So again, there's a scientist or student or some sort who stained the cells, and now they can see them. And so you'll see it has around 3 to 7 lobes, and their main job is to be active phagocytes. So they'll run around eating other cells. These are typically found with a short-term or acute infection. So again, medical technologists or clinical lab scientists, or maybe even pathologists, their job is to look at these blood cells under a microscope, get a rough count, and that helps the medical doctor to come up with a diagnosis. So there's around 3,000 to 7,000 of those in this millimeter cube of blood. We have a cytophils, which are red coarse cytoplasmic granules and are bilobed and stained red. These kill parasitic worms and increase also during allergic attacks. They will phagocytize antigen antibody complexes, so they help to digest and eat things that aren't supposed to be there as well. And and they also inactivate some of the inflammatory chemicals. So anything that kind of stimulates that, they'll respond to. We also have basophils, which are granules that contain histamine. They're vasodilators, this is the kind of stuff that kind of gives you that inflamed response that you don't always appreciate when you have allergies and so forth. The cytoplasm has a few large blue-purple granules, kind of a U or S-shaped nucleus with constrictions containing dark blue. So again, this is what you're going to see under a microscope after being properly stained. So those are the granulocytes. Now the agranulocytes means you don't see the granules. So the granulocytes would have been like the purples and the blue spots. Let me go back to the previous slide. You can see the little dots, those are granulocytes. So in addition to the lobes, which is the nucleus, the big lobes are the nucleus, the little dots are the granulocytes. So those are granulocytes. Basophils would be another one. Again, that's the kind of the histamine allergic kind of inflammation response, vasodilators. Then we have the agranulocytes, and you can see the basophils is pretty small in numbers, but the lymphocytes are part of the formation of antibodies. So lymphocytes have a clear, they don't see the granules, and their nucleus is actually rather large. So you can imagine having these large nucleuses are important for all the different proteins that the immune cells make. So again, there's lymphocytes that they produce antibodies, and then T lymphocytes that help fight with tumors and viruses, and activating B lymphocytes. So these are the cells that are responsible for making antibodies. While monocytes, again, are active phagocytes that become macrophages in tissues. So they're more involved in long-term cleanup and increase in numbers during chronic infections. A good example of chronic infection is tuberculosis, which is obviously a pretty serious lung disease that would cause people to be sick for long periods of time. I often think about this as more in the cowboy days, but tuberculosis also exists around today. So again, they're abundant gray-blue cytoplasm, dark blue, purple nucleus, and often kidney-shaped. Platelets represent 250,000 to 500,000 platelets. They're essentially irregular shaped cell fragments that stain deep purple. And they're needed for normal blood clotting, initiating clotting cascades, and clinging to broken areas. So platelets come from megakaryocytes. They're just literally fragments of the cell that have been broken apart, floating around in their blood, and then they will stick together along with fibrogen to form a blood clot, as you'll see. So here is a totipotent cell that becomes committed. So the stem cell becomes committed. Let's see if I can move that out of the way a little bit. So this totipotent, and then it becomes pluripotent. So initially it can be any kind of cell. Some of them will become lymphocytes, as you can see the arrow going down, so they'll become a lymphocyte, which of course helps in making, which is an agranulocyte, so it's important for making antibodies. And then we have stem cells that become the rest of the types of cells, and that includes erythrocytes. So you can look at the far left, you can see the erythrocyte does have a nucleus, and the nucleus is present as hemoglobin is being made. It'll be called an erythroblast when it's still not a fully mature red blood cell. And then eventually that nucleus is either ejected or broken down. I'm not sure which one is exactly the case, or maybe both. It eventually becomes that concave shaped erythrocyte that is full of hemoglobin. Unless, of course, it's a situation where somebody has sickle cell anemia, and then the red blood cells don't have the proper shape. So they don't have that nice concave shape. And so those can be situations that can cause a problem. And again, that's related to some genetics that affect some of the proteins and the hemoglobin that cause the shape to be strange, as I remember. Some cells become like this huge megakaryocyte. The megakaryocyte then breaks off as fragments of platelets. So this be literally breaking off segments of it. Then you have some that become neutrophils and monocytes. And then, of course, you have your basophils and the cytophylls. This all takes place in bone marrow. And so here's another picture showing a cytoblast, which is a stem cell. At this point, it becomes pluripotent if it's a hemocytoblast, because it's designated to become some type of blood cell. So we have our lymphoids that become our lymphocytes, and then we have our myeloids that become our stem cells, which again includes erythrocytes, platelets, basophils, eosinophils, and monocytes. Found in the red blood cells, and so forth. I actually had a cancer. When I was about 2006, 2007, I had lymphoma. So it's actually caused by lymphocytes. Like in my case, it was a large B cell lymphoma. It was actually a non-Hodgkin's lymphoma. There's probably around 25 or so, as last I recall, non-Hodgkin's lymphomas. And I actually had atypical Burkitt's lymphoma. It's actually a lymphoma that's typically found by people in Africa. So I actually had the atypical because it was found, of course, in the United States and at an older age. So later along my right neck, I had a bump grow. And if you ever have a situation where you have a bump that's growing rapidly and kind of hard, that's a sign of something that really needs to be checked. Don't be scared and not go to the doctor. Get that bump checked, because it could be a cancer. And if you catch it early, you can survive. I'm a living testament of that. The difficulty about my typical kind of cancers is you had to use a very aggressive chemo. One of the most aggressive chemos, even had spinal injections and all sorts of things. I went through the whole thing, bald headed, bald everywhere, mouth sores, the feelings of nausea. It was really awful, but I survived, and so forth. One of the things they did when my blood cells were getting low is they would give me hormones like erythropoietin to stimulate me making new red blood cells and white blood cells, because I wasn't producing enough of it after the chemo was hit. So this would happen every now and then. And it was amazing when I'd give myself this injection at night, I would literally feel my bones radiating. It was that intense that the bone marrow was almost like this painful feeling I could feel in my skeleton, where the red blood cells were being, and other blood cells were being kicked into overdrive, particularly white blood cells. That's what they were worried about. Because you'd get the chemo would knock down your immune system so badly that they're afraid I would get sick from something else. Here's a picture showing you the blood cells forming in the bone marrow. And so you can see your neutrophils being formed and matured, and then ultimately going into the blood. You can see the megakaryocyte on the upper left side. You can see it has extensions, almost looks like the blob. It says fragments of megakaryocytes break in and break off to become platelets in the blood. Again, that's important for blood clotting. You have your red blood cells. You can see it's a stem cell, and then that becomes um, it says the reticulocyte is expelled. Um, so that's expelling the nucleus, and so that's going to become the red blood cell. Once that nucleus is expelled, it'll become a erythrocyte. So right now it's a reticulocyte until that nucleus is expelled. And then you can see it just floating around in the capillaries. Those are capillaries right there. Shows you macrophages. And remember, a macrophage is kind of like an amoeba. It'll just be kind of moving around and will engulf a particle of some sort and eat it. Here's a megakaryocyte here, breaking off fragments. So you can see the platelets are a lot smaller than the erythrocytes. Again, these are just fragments of the giant cell. And anyhow, so the main function of erythrocytes is to carry oxygen. It's bioconcave. It's anucleus. It contains very few organelles and outnumbers white blood cells by 1,000 to 1. Here's what a hemoglobin looks like. It's an iron-containing protein that strongly binds but reversibly to oxygen, unlike carbon monoxide, which can attach to the hemoglobin and not let go. The hemoglobin contains an iron structure. So each hem group contains a porphyrin ring with an iron atom in the center. So you can see how the iron is attached to the covalent bonds. Now each hemoglobin actually is made up of 4 molecules. There's an alpha chain, a beta chain, and there's 2 of those that stick to each other. And again, there's an iron with each one of them. So each complete hemoglobin has 4 irons subunits, 4 separate hemoglobins that stick together. And if I remember right, as mentioned above, each erythrocyte has 250 million hemoglobins, more or less. So you're talking over 1 billion sites to hold on to oxygen. Now you can see this is a graph that I want you to learn, and would like you to, obviously you're going to have this for your exam. So it may not be a bad idea to draw it out and practice this a little bit and think about it. Good chance that this will be part of your final oral exam as well. But you're obviously going to take in iron from your diet, and iron is critical, particularly if you're not eating enough protein, red meat, and things like that, or have other good sources of iron. You'll need to take iron supplements. Some people that are anemic, and women tend to be more anemic than men, need to make sure that they add extra iron in their diet often. But again, talk to your healthcare provider to see if you're in that kind of condition. Iron is actually going to be absorbed in your intestines through active transport. So that means it's going to require ATP. That iron will then be moved around with the help of transport proteins. Remember how we talked about how there's transport proteins in your blood plasma, for example, will have transport proteins. In this case, it's called transferrin. So transferrin will move iron to the bone marrow. And then the bone marrow will, through, I'm sure, various enzymes and pathways and stuff like that, will attach to the hem, making hemoglobin. Looking at number 5 right now. The liver will store excess iron as ferritin. So when it's not being utilized, if you go down to number 4, ferritin will be the way the iron stores excess iron. So the red blood cells and the hemoglobin will live about 7 months or so, running around collecting oxygen, doing its job. And when is at some point the body will designate as no longer being good. I'm not sure what the trigger will be, but then it'll go to the spleen where it'll be destroyed. And then the spleen will convert the hemoglobin to bilirubin. Bilirubin is important for going into bile. If you remember, bile is found in the gallbladder, or you might know, bile is stored in the gallbladder and is important for lipid absorption. So the bilirubin, again, can be used for bile salts. And so if you go, it has two different mechanisms. So it can either go to the kidneys, and the excess bilirubin will be released in the urine or the feces, and I'll give it a little bit of that urine that's yellow color, the bilirubin. Or the bilirubin will undergo some more metabolism in the liver, where the gallbladder is located nearby, and then the bile will go into the gallbladder and then be part of the bile duct that will go into the intestine to help in absorption of fats. So there's a thing called bile salts. So instead of having a big droplet of fat running around in your intestines, the bile salts will attach and make big oil droplets into small oil droplets. Ultimately, you'll digest and absorb the fats, but some of that bilirubin will be wasted as feces if it goes back into the intestine, or as urine if it gets filtered by the kidneys, of course. So again, a mature erythrocyte is not going to be able to divide. It doesn't have a nucleus anymore. So after 100 to 120 days, it will be let go, so to speak, and then go either to the kidneys and be lost as bilirubin there, or go to the liver and be made into bile salts that go into the gallbladder. But again, the spleen and the liver are important for that, reclaiming that kind of stuff and or getting rid of it. And then your bone marrows and the hematocytoblasts will continue making fresh red blood cells. Again, erythropoietin is the hormone responsible for helping trigger the bone marrow to making more erythrocytes. So if you start exercising or working in high altitudes, your kidney is going to release more erythropoietin because it's going to notice that the oxygen levels are pretty low in the blood. So that's what's triggering it. So the kidneys can monitor your blood, and that makes sense because your blood is constantly being filtered by the kidneys. So again, if there's homeostasis, your body's constantly trying to maintain the amount of oxygen that you need. So if you stop exercising, your body's going to make less erythropoietin or less red blood cells, less erythrocytes. Thus, a less erythropoietin hormone is going to be formed. If you exercise a lot more and your cardiovascular system is under more demand, your body's going to detect that there's lack of that oxygen, and then it's going to go on and produce more of the erythrocytes and so forth. Hemoglobin binds to the oxygen in the heme group, as I mentioned. And then some of the globulins will bind with carbon dioxide, which is a protein also. Bone marrow makes about 2 million red blood cells per second. So that's incredible. So I wonder what I was doing when I was taking that erythropoietin. It must have been dramatically higher. While still in the marrow, the immature cells divide many times and produce hemoglobin. When the hemoglobin levels reach 30% of the cell volume, the cell organelles, including the nucleus, break down and the cell enters the circulation. Each red blood cell lives about 120 days and then breaks down. The spleen serves as the reservoir for old blood cells that have been squeezed and ruptured, and the cell remnants are then broken down by macrophages. Remember the macrophages? They're like amoebas that'll run around and eat it. Besides producing leukocytes and erythrocytes, the stem cells in bone marrow will produce the megakaryocytes. Again, megakaryocytes are fragments that break off into platelets that allow for clotting. The swelling of sticky platelets release clotting factors that activate other platelets to initiate clotting, and thus allowing the blood cells stick together and prevent you from dying. Here's a picture that is easy to learn. You can see that this is a negative feedback loop. When the oxygen levels are low, we have lower levels of oxygen, the kidneys will release more erythropoietin. And that erythropoietin will then trigger the bone marrow to stimulate and make more red blood cells. And then once the oxygen levels are where they need to be in red blood cells, you'll be back to normal. Now, this isn't always the case. And the bone marrow is important for us to make red blood cells, but actually, the fetus's liver and spleen are also important for blood cell formation. So the body of a fetus is more portions of it are involved in blood cell formation, i.e., the liver and the spleen. Fetal hemoglobin actually differs from the hemoglobin after birth. The reason why is the fetal hemoglobin needs to be able to take the oxygen from the mother. So it's actually it has more affinity for oxygen. So the hemoglobin of the fetus can grab onto oxygen out of the blood, cause a concentration gradient that'll pull the oxygen from the mother's hemoglobin to it. Otherwise, the baby or the fetus won't get enough oxygen from the blood of the mother. So that's why the hemoglobin is different for fetuses than for the adults. Going back to another slide I have, we're talking about respiration. We'll get back to the blood cells. So we're jumping back to the respiration notes for just a split second, and I'll catch up on some of these other graphs. Oops. We're on circulation. So as you recall from the respiratory, now we're looking at the respiratory notes for a moment because it's related. You can see the heme group again, where they have the iron. I already mentioned carbon monoxide and so forth can attach to the hem group irreversibly. Well, the hemoglobin, as you might recall, remember each one of these red blood cells, the hemoglobin is going to be composed of 4 units. So each one of these hemoglobins can hold 4 oxygens. And then there's also some oxygen that's saturated in the blood plasma, particularly near the lung capillaries. So there's a lot of oxygen diffusing into the blood. Some of it's going to attach to the hemoglobin. Some of it's going to dissolve into the blood plasma, the watery area. And when it does so, what happens is all the hemoglobins hold on to the oxygen strongly. It's after the oxygen, some of that oxygen leaves the blood plasma and drops in levels, that's when the hemoglobin will begin to release the oxygen from the 4 subunits. So again, when it's near the lungs, the hemoglobins will grab onto the oxygen, each one of those hem groups with the iron, but there's also saturated and dissolved oxygen in the blood. And so at that moment, the hemoglobin holds on to the oxygen strongly. And then as it travels through the body, it's going to start reaching tissues where the oxygen that's in the blood plasma is going to diffuse and leave the blood plasma. And in doing so, it's going to change the amount of oxygen in the environment. And then that will cause the hemoglobin to liberate and release some of its oxygen from the hemoglobin. It may, in general, only release one or two of these oxygen molecules from any given hemoglobin. It's only under extreme situation where your body's really starved for oxygen that it will liberate the rest of it. So normally the blood can actually circulate. The red blood cells can actually circulate in your body and hold on to most of the oxygen, maybe holds on to all but one, so or two, and then it'll come back and refill, so to speak, in the lungs. It's only under extreme situations where the binding would cause the rest of the oxygens to let go. So if you look at this graph here, you can see when the amount of oxygen that's dissolved in the blood plasma, the watery portion, is high, the hemoglobin holds on to it tightly. That's what you see with the graph. The hemoglobin's holding on to it 100%. And if the oxygen dissolves down to around 60% in the blood plasma, it's still holding around 90%, the hemoglobin is. You don't really see a drop off until about 40% of the dissolved oxygen in the blood or whatever is gone before you really see it now liberate more oxygens from the hemoglobin. So you got to lose a lot of oxygen in the blood plasma for the unbinding of the oxygen of the heme group takes place. So when the blood circulates through the body, it releases on average only 1 in 4 of the molecules of oxygen it carries. So the hemoglobin keeps 75% of its oxygen in reserve for peak demands. If a tissue is starved for oxygen and its local saturated oxygen and dissolved oxygen falls below 40 millimoles of per mercury, or whatever that symbol is, the hemoglobin will release its reserved oxygen. So again, the take-home message is the hemoglobin holds on to most of the oxygen until the tissues or the fluids themselves, because remember, blood plasma is considered a liquid tissue. When that oxygen level it's actually in the fluidy part dissipates low enough, that's only when the oxygen hemoglobin will release its oxygen. So there's a relationship between the saturation of hemoglobin and the oxygen. That's what you see in those graphs there. Oxygen binding to hemoglobin is influenced by positive cooperativity. That is, when the first molecule binds, when the first oxygen binds to the hemoglobin, it makes the rest of the hemoglobin bind on to oxygen rapidly. So that's the other thing. So it's actually easier to cause the hemoglobin to become saturated with oxygen. pH also influences how well the oxygen will hold on, or the hemoglobin will hold on to the oxygen. In a more acidic environment, when the pH falls, there's more hydrogen ions, because remember, acids and pH are mostly due to hydrogen ions. The hemoglobin will decrease its affinity for oxygen, liberating more oxygen to its tissues. That's usually a good thing. But if your pH really became low, then your hemoglobin can no longer hold on to oxygen and you would die. So if you were to shoot up straight hydrochloric acid into your blood, not only would it denature the proteins, it would also cause your hemoglobin not to be able to hold on to oxygen. Your body would obviously have buffers to try to fight it, but if you're talking large amounts. So again, in an acidic environment, the hemoglobin will liberate some of that oxygen. Another regulator of hemoglobin function is 2,3-biphosphoglyseric acid. In red blood cells, BBG, which is biphosphoglyseric acid, combines with deoxygenated hemoglobin and causes it to have a lower affinity for oxygen. The result is that hemoglobin releases more of its bound oxygen to tissues than usual. So if a person goes to high altitudes or starts exercising, the levels of biophosphoglyceric actually goes up, and allows the hemoglobin to release more oxygen when it's needed. So trained athletes will have more of the 2,3 biophosphoglyceric acid, or if you train at high altitudes, you'll have more of it. And that's a benefit because that means more of that oxygen will leave the hemoglobin and go to the tissues that need it. So well-trained athletes can take up, their bodies are more good at, or more efficient at giving and releasing that oxygen. Some structures like myoglobin, these are, as you can see, it's kind of like the name hemoglobin, but myoglobin, globin being the protein, is found in muscle cells, and it's an oxygen binding molecule that takes up one molecule of oxygen. So this is one of the reasons why your muscle has that red appearance. It has a higher affinity for oxygen than hemoglobin, which makes sense because you need to be able to pull the oxygen away from the hemoglobin and put it into the muscles that needs it. So again, myoglobin is going to have a higher affinity for oxygen, causing a concentration gradient that pulls the oxygen away from the hemoglobin and puts it into the muscles that need it. Diving animals such as whales and seals and things like that have unusually high concentrations of myoglobin in their muscles. This allows them to take even more oxygen from the blood and allows them to stay underwater for longer periods of time. So they're more efficient at holding on to oxygen in their tissues and pulling oxygen out of their blood. Even non-diving animals' muscles called for extended periods of work frequently have more myoglobin than muscles used for short periods or intermediate periods. So in other words, athletes that are endurance athletes will have greater amounts of myoglobin than sprinters. So not all hemoglobin is created equal. As I mentioned earlier, fetuses have a different type of myoglobin. They have 2 alpha globulin chains and 2 G globulin chains instead of beta. And that results in a greater affinity for oxygen. So human fetal hemoglobin has a much greater affinity for oxygen than adult hemoglobin, which has alpha globulin and beta globulin. So the G globulin helps it to have more oxygen affinity and able to pull the oxygen from the mother's blood. We call that a concentration gradient event that help to do that. Again, if you work at high altitudes, you might change your hemoglobin a little bit too and increase more myoglobin. So all sorts of things like that happen. So if you look at the mother's maternal hemoglobin and its affinity for holding on to oxygen, you'll see that it'll give up oxygen faster in a oxygen saturation. Remember the oxygen saturation that's in the blood, the PO2? Well, if you have around 40% of that saturated oxygen, it's going to liberate it more. While in the case of a human fetus, it's going to be even greater. So the green one, so you have the purple, green, red, and blue. So you can see the purple gives up its oxygen more easily when there's less oxygen in the blood, saturated oxygen in the, when I say blood, I mean the blood plasma, not the hemoglobin. So again, the hemoglobin is going to liberate oxygen more easily than the human fetus, which has that greater affinity to hold on to it. Llamas, which are high altitude animals, have a hemoglobin that can really hold on to oxygen, even in these high altitude environments. So their hemoglobin has a greater affinity for oxygen than your hemoglobin does. And then the structure that has the most affinity for oxygen, myoglobin, because again, your muscles are going to need to pull that oxygen from the blood. And so it's going to have the greatest affinity for oxygen. Here's your Napoleon Dynamite quote. So one of the things you should do is go watch the movie if you haven't seen it, but you can tell it's a little bit dated now. So here's our llama, and as you can see, it has the greatest affinity for oxygen, their hemoglobin. And as you can see, Tina, you blankety blank, come eat some dinner. Tina, eat the food. Remember when Napoleon Dynamite did that? It's pretty funny. Anyway, and he also tried to feed the llama ham. This was quite hilarious when I first gave this lecture, of course. Now, remember, carbon dioxide is actually not going to attach to the hemoglobin. Carbon monoxide would, and irreversibly in most cases. So carbon dioxide is produced by the tissues during cellular metabolism. Remember, when we break down sugars and lipids and so forth, we're going to break it down into CO2. You're going to breathe in oxygen that helps the mitochondria, that's where this is taking place, to make ATP and ultimately carbon dioxide, the waste product. Most of the carbon dioxide will be transported in the blood as a biocarbonate, which is a good thing because it helps in maintaining that pH of our blood. Carbonic anhydrase is actually what's going to convert carbon dioxide with water into biocarbonate and a hydrogen ion. So the hydrogen ion and biocarbonate will balance out. And that's important for other parts of your body in controlling blood pH or tissue pH and so forth. The carbon dioxide will transport in the blood as biocarbonate, H2CO3. This associates and biocarbonate ions enter the plasma in exchange for chloride ions. This actually allows for some of the, let me just to back up a little bit. This is important. Remember when I told you about how the mucus is formed in different organs and lungs in the previous lecture? I talked about it in regards to lungs. And if you have cystic fibrosis, that can be an issue. Well, this chloride and these kind of things that are found in the blood plasma that are floating around are important for helping make this mucus watery, whether it be in the lungs or the gut. And we'll get into that a little bit later in another class. So basically, it's not really, for the most part, CO2 running around. It's going to be in the biocarbonate form. And again, the enzyme responsible for making that biocarbonate is carbonic anhydrase. Then when this blood reaches the lungs, the carbonic anhydrase, because remember, enzymes are reversible. So they can go to making biocarbonate, and then they can be reversed to make carbon dioxide and water again. And so when it reaches the lungs, the carbonic anhydrase will convert the biocarbonate back into CO2, and then that will diffuse into the alveoli or the alveolus. And then you'll breathe out that CO2. Now remember, the CO2, this is also related to the CO2 levels in blood. Because remember, some of that CO2 is going to be saturated in the blood, just like the oxygen. So some of the CO2 will combine with hemoglobin. In the lung, the CO2 and the biocarbonate reactions are reversed, also catalyzed by carbonic anhydrase. Carbon dioxide diffuses with the blood plasma into the alveoli and is exhaled. As that carbon dioxide in the blood falls, biocarbonate will be converted to carbon dioxide. So let me make sure you understand that. So you have blood transporting carbon dioxide as biocarbonate, but you also have dissolved carbon dioxide in your blood. And so when the dissolved carbon dioxide starts to leave and go through into the alveoli, the carbon dioxide levels in your blood drop, saturation levels. And as a result, carbonic anhydrase and so forth and probably other triggers will start converting biocarbonate back into CO2. And then that CO2 will also be released into the alveoli for you to exhale it. Remember, CO2 levels is actually what causes you to inhale and exhale. High levels, particularly, are influenced by that. High levels of carbon dioxide really triggers breathing rates, even though low oxygen also does. So I think that's a pretty good area to stop. Next time we get back together, we're going to talk about anemia and blood loss and hemostasis. How does the body stop you from bleeding to death? So that's what we're going to focus on next time. Let's see if there's any quick little videos that I can throw in there that might help to explain some of this. All right, so this little video might help you to understand what I was trying to explain a little better. Here you can see your hemoglobin. It's, again, a quaternary structure. It's made up of 4 polypeptides. You can see the hem group in the center, and you got some oxygen attached to it. When it's near the lungs, the partial pressure of oxygen is high, and thus the hemoglobin is going to hold on to it. But when hemoglobin reaches the muscles, the partial pressure of oxygen in the blood is going to be lower, and so the hemoglobin is going to release more of its oxygen. Also, remember the myoglobin is present in the muscles. It's going to help pull the oxygen out of the blood.
A factor influencing the production of oxyhemoglobin is the partial pressure of oxygen. The partial pressure of oxygen determines the number of oxygen molecules that can bind.
So you can see the oxygen molecules on the outside that's floating in the blood. That's the partial pressure. And then again, there's the hemoglobin that's holding on to oxygen too.
Bind, oxygen loading, or dissociate, oxygen unloading from hemoglobin. Blood in vessels coming from the lungs is very high in oxygen partial pressure, so saturation is high. This is because oxygen binds to all available sites in hemoglobin and forms oxyhemoglobin. Blood near skeletal.
Notice how they called it oxyhemoglobin when it's holding onto the oxygens. That's something I didn't mention earlier. And then when it reaches the tissues and the muscles far away from the lungs, you notice the saturation of oxygen around the blood is now dissipated, which is going to cause the hemoglobin to liberate, the oxyhemoglobin to liberate its oxygens.
Muscle cells is very low in oxygen partial pressure, so saturation is low. The higher partial pressure of oxygen bound to oxyhemoglobin causes the oxygen to detach, and it is unloaded to the tissues. Oxyhemoglobin saturation is affected by blood pH values. Increased metabolic acids, like lactic acid and carbonic acid.
Remember, when exercise, you're going to build up lactic acid from exercising intensely. That'll actually cause the oxygen to be released from the hemoglobin at a higher rate. And again, we call this the Bohr effect. And so here you can see hydrogen ions, because remember, hydrogen ions are important for making, the more hydrogen ions you have, that's when you lower pH, right? So that's an acid. Hydrogen ion means a hydrogen atom, the nucleus, without an electron around it.
Enhanced dissociation of oxyhemoglobin and the unloading of oxygen. This effect is called the Bohr effect. Lowered pH increases the unloading of oxygen from oxyhemoglobin, thereby making oxygen available for actively metabolizing cells. In contrast, elevated pH values increase the affinity of oxygen to hemoglobin, thereby lowering the unloading of oxygen to tissue cells.
All right, so as I mentioned before, the next time we'll get together, we will talk about we're going to talk about hemostasis and a few other things. Let me just double check one last thing and then I'll call it a wrap. Okay, one more video for us. Obviously, you know about hemoglobin transporting and erythrocytes transporting oxygen.
Oxygen from the lungs to body's tissues and removal of carbon dioxide in the reverse direction. Erythrocytes lack most of typical cell structures. They have no nucleus and almost no organelles.
Remember, that's the mature one. Before that, the red blood cells was an reticulocyte or something like that, involved in when the development was taking place. And so then that nucleus gets kicked out in those organelles. Let's move along a little bit. We already see it's shaped like a donut. Here's the hemoglobin. Again, it's going to be 2 alpha chains and 2 beta chains in adult humans, while gamma chains in fetuses.
Polypeptide chains, each of which is bound to a red pigment molecule called heme. Heme binds oxygen to a ferrous iron in its center. Thus, a molecule of hemoglobin can bind up to 4 molecules of oxygen. Binding of oxygen is a cooperative process. Binding at one site changes the protein conformation in a way that facilitates further binding at other sites.
Yeah, so anyhow, that's where the heme comes along. Now let's move along a little bit. So we're talking about oxygen partial pressures. So again, there's going to be high partial pressure of oxygen in the lungs and low partial pressure of oxygen in your tissues, and that'll cause the loss of oxygen. But what about carbon dioxide? Anyway, let's keep watching.
Associates in tissues where its pressure is low. While hemoglobin is responsible for transport of most of the oxygen, it only carries a small portion of carbon dioxide. Carbon dioxide binds to the polypeptide part of hemoglobin and not the heme, but its
So you can see carbon dioxide can attach to the hemoglobin, just not to the heme group.
binding changes the conformation of the molecule and decreases its affinity for oxygen. In other words, the two gases compete for binding on hemoglobin. Oxygen binding is favored in the lungs, while carbon dioxide binding is more favorable in tissues.
Well, that makes a lot of sense, right? Because you know that when the lungs have lots of oxygen, while the atmospheric carbon dioxide is low, and so when the blood reaches the lungs, it's going to want to liberate that carbon dioxide and take in oxygen.
The majority of carbon dioxide is transported in the blood in the form of bicarbonate ions. Conversion of carbon dioxide to carbonic acid, which dissociates into bicarbonate and hydrogen ions, is catalyzed by an
So again, the enzyme responsible for turning carbonic acid into bicarbonate hydrogen ions is carbonic anhydrase.
enzyme present in red blood cells called carbonic anhydrase. Bicarbonate ions then diffuse out to the plasma to be exchanged for chloride ions, while hydrogen ions bind to hemoglobin.
So remember, the hydrogen ions and the chloride are going to be attracted to one another because of the positive and negative, while the bicarbonate is floating around in the plasma after that chemical reaction takes place. We're going to have high amounts of carbon dioxide still associated with the attached to the hemoglobin until it reaches the lungs. And then when it reaches the lungs, this carbon dioxide is going to leave and go into the lungs through the alveoli and the thin membranes. And then this bicarbonate is going to be converted back into carbon dioxide to also be released.
Hemoglobin that has released oxygen. When red blood cells reach the lungs, the reverse happens. High oxygen pressure favors its binding to hemoglobin, which releases hydrogen ions and carbon dioxide. The same carbonic anhydrase then converts bicarbonate and hydrogen ions back to carbon dioxide to be breathed out.
All right. So anyway, that finishes up our discussion on blood outside of hemostasis. And we'll have a little bit more about the circulation and how it's regulated. And that will probably wrap up these two sections, and then we'll be on to discuss digestion and kidneys, and then maybe some miscellaneous reproductive stuff. That'll hit most of the major systems. Digestion, I think, will be fun to talk about. But let's go ahead and we'll finish up another lecture or two on blood and how we stop ourselves from bleeding to death, hemostasis, and so forth. Anyway, thanks for watching. Please write a summary and turn that in. Take care.